Okay, Assalamualaikum warahmatullahi wabarakatuh, and very good afternoon to Dr. Puat and all members that have been watching this presentation. So today I'm gonna present about the proteomics applications. There will be multiple application of the proteomics and also the contribution to the current research and science. So before we begin, my name is Muhammad Adham bin Bakri, and my metric number is 200737. So without further ado, I will present about the proteomics applications. So let's go for the first uh, our points today. So the proteomic application and contribution, the first is actually about the production of a biomarker from proteomics techniques. So actually biomarker are one of the proteomic techniques required to make a biomarker. So the second one is the drug discovery. The third one is the proteomics in nutrition research. The fourth is the diabetes research. And the last one is protein mapping techniques that also require the application of proteomic techniques. And we will discuss it later. So let's start with the first application that we get from proteomic, which is the production of biomarker. So actually, what is the biomarker? Biomarker is actually one of the we call indicator, or it also called the reliable indicator, which is this reliable indicator actually comes with the four types, which is predisposition biomarker. The second one is diagnostic biomarker. The third one is prognostic biomarker, and the fourth is a predictive biomarker. So actually, there are many types of the reliable indicator, or we call it as biomarker. So every type have a specific functions, or we can say has specific uses. So actually, for example, like diagnostic biomarker is usually used for application when we want to diagnose or analyze the protein that we want, or we can say it's a protein of interest. For example, when you want to find the protein that affect the height of the plant, we can use diagnostic biomarker, okay, to find the our gene of interest in the plant. So other than that, the predictive biomarker also can be used to predict the detection of but detection of the other foreign protein, of course specifically because it's very, very specific at the molecular level. So in general, biomarker also has functions. So the function of the biomarker are firstly about to identify the biological activities and biological process. So instead of knowing gene, specific gene, it also can identify the biological process activities when the products, or maybe the starter things for the reaction can be fully recognized by the biomarker. So we can analyze the quantity of the products and the byproducts. So from that, we can know there's a biological activities and biological process. So that's how biomarker identify the biological activities. The second one is detecting specific gene on the genetic level. Like I say before, is when we talk about the specific gene detected by a biomarker, it actually usually used in agriculture. Actually, the scientists usually use it to detecting the gene that control that some of the part of the plant. For example, maybe the gene that control the height of the plant. Usually in agriculture, they want to modify the height of plant. Sometimes they want to be lower, to easy for collecting the food, the fruits, and sometimes they want to be higher to have the some advantages. So from that, scientists usually use biomarker to detect the specific genes that they want to maybe delete, change, or cloning the genes that are of interest to make the specific level. So in the genetic level, detecting the specific gene are really useful by using the biomarker. Okay, so the last one is the function of biomarker also capable of analyzing many proteins simultaneously. So, for example, biomarker also being used in a protein microarray and protein spectrometry, where these two types of techniques in proteomic usually use biomarker to analyze, to quantify the proteins in a specific way simultaneously. So biomarker are very have specific function, and also usually for the proteins. Okay, so let's move on to our example of the protein application of biomarker. So actually, as I said, example usually biomarker being used in the agriculture field, where scientists use to detecting the specific gene in the plant. But in this example that I give here, is actually an example for in the medical field. So in the medical fields, the biomarker usually also contribute to make the cancer biomarker. For example, like Roma and 5. So Roma is a stand for risk of ovarian malignancy algorithm, which is this biomarker improve the performance in predicting the ovarian malignancy in the patients. So this is very good biomarker since it can use to the live organism, especially for a human that and doesn't have a side effect. So this help the doctor to improve the performance in choosing which side the ovarian malignancy occur in the patient. So this make the doctor have a shorter time to treat the patients. The second one is the 5 with PHI that stand for prostate health index. Though this biomarker improve the accuracy of prostate cancer detection and avoid unnecessary biopsies. So this means that 5 biomarker can detect the prostate cancer location accuracy, also help the doctor for avoiding the unnecessary steps of the treatment, because this sometimes without the 5, the doctor have a hard time to detecting the specific location of prostate cancer, and this make the doctor to make more biopsies and steps that should be unnecessary, but you still need to try and error to in order to locate the actual prostate cancer location. So with DeFi, this avoid the doctor to use unnecessary biopsies and also reduce the cost for the patient to pay for the treatment. So it's very good in terms of the medical and also the economical. So like you say that biomarker are very useful in the application of proteomic and have contributed a lot. So the next is about the drug discovery in a proteomic, using the proteomic techniques. So how the drug is being discovered by using proteomics techniques? The first one is actually they facilitate and accelerate the discovery process, actually by using the target identification, and useful for drug study, action study, toxicity, resistance, and efficiency. Usually, a drug can be studied by using the proteomic techniques to discover its actions, how the drug acts, how much the toxicity level for the every drugs that have been discovered or has been tested, the resistance of the cell metabolism to the drugs, and the efficiency of the drug itself towards the cell's metabolism. So all this kind of characteristic and discovery can be made by proteomic techniques, which are the target identification techniques by proteome. And then other than that, the target identification is the first step in the drug discovery process, and the identification and also early validation of a disease modifying target are essential step in the drug discovery pipeline. So actually in the drug discovery, they also have a genomic methods that play important role to come up with the drug discoveries info. But the most crucial and the very critical part that really need that really contribute in drug discovery actually proteomic methods. So this come up with a proteomic method is more contribution from the genomic method. So why? The reason is the proteomic method is more contribute than the genomic methods in the drug discoveries, actually because the gene expression profile is not indicative of the protein expression. So this does explain the genomic is capable of presenting cellular event only to the transcriptome state. So unlike the proteome. And therefore, most of the investigation usually will be focused on the proteomic studies, which is we evaluate every protein that we want or protein that we interest, and then they are post-transformation in the large scale. So PTM also contribute a lot when you want to analyze a protein, because some protein will be change the shape or modify, like PTM's process that change protein structure and in large scale. So using proteomic technique also enable you to do that in the large scale. And in the complex drug discovery pipeline, usually very specific drug discovery, various MS-based proteomic approach also been a common objective for the drug target discovery. And also this enable us to the study of the drug target interaction. So like the selectivity and the specificity of the drugs for the specific cell metabolism, right? So maybe the drugs have a side effect for the human, but not for the animals, but maybe have side effect for the animals, but not for the plant. So that's how we detecting. The idea is that other than that, it's also we can monitor the drug activities by the proteomic techniques. Checking or analyze the protein solution or concentration using the proteomic method, many type of experiment, which is we can analyze also the drug activities and elucidating the mechanism of action of a new drug. So maybe sometimes from the analyze, you can make a new drugs based on proteomic techniques also. Okay, so other than those application, there is some very interest application or the contribution of proteomic, which is in the nutrition research field. So actually, proteomic also contribute in the nutrition research. So in discoveries in nutrition research, including profiles and characteristic of dietary and body protein. For example, usually that happen in us is digestion, absorption, and metabolism of the nutrient. So all these activities of the intestine can be analyzed by the proteomic techniques by nutrition research. So from that, we can make our own, not own, you can come up with the idea in a healthy lifestyle. Like I said, it will be including profile and characteristic of dietary and body protein. So with the molecular level information, you can make the more healthy lifestyle based on scientific info. Other than that, is the function of nutrient and other dietary factor in growth, reproduction, and health. So, like I said, if you have the molecular information, so your specificity of dietary will be more good. And sometimes the biomarker of the nutritional status and disease and individualized requirement of the nutrient, which is usually use sometimes biomarker to detect a specific nutrient in order to obtain the nutritional status and or to find out the disease. So the solving major nutrition associated problem in human and animals, such as obesity, diabetes, cardiovascular disease, cancer, aging, and intrauterine fetal retardation. So that's one, not one, more than one actually of the chronic disease that have been countered by using proteomic techniques. Not the proteomics alone, but maybe have some techniques to contribute to, but the main contribution is the proteomics in the nutrition research that help in fighting those chronic disease. So, specifically in the diabetes research, describing the healthy tissue and to unravel the complex pathophysiology behind the disease also being apply to the proteomics actually. For like effect on the protein induced by the different treatment have also been investigated using proteomic approach. That's how the diabetes research being performed to increase or avoid diabetes research by using proteomics. So other than that, proteomic also contribute into the protein mapping in disease detection. So usually protein mapping is one of the type of techniques that are used in proteomic to detect disease. Quite familiar right now in the scientists. So usually the idea is the proteome is dynamic and constantly changing in response to a cell's or organism environment. So the variety of protein present and their relative abundance change in response to such phenomena as stress, disease, and drug treatment. So from that, you can have sample and make a protein mapping to detect a specific protein. Like, for example, classical 2 dimensional electrophoresis technologies can be used and actually can analyze the protein content of the cell and tissue as well as body fluids such as plasma or urine in combination with mass spectrometry. So that's some of example that you can, or the analysis technique in proteomics you can use in protein mapping to detect disease that currently undergo in our patient or ourselves. So nowadays, scientists, doctor usually use such experimental techniques to make the specific treatment and improve the medical field contribution. So other than that, also the proteomic also make the protein profiling of replicated isolate. This one of the specific example that I found in the PubMed website, which is a trusted article, which is I got the insulin producing beta cell in isolate of Langerhans is central in the glucose homeostasis. By its dysfunction is part of the pathogenesis of both type 1 and 2 diabetes mellitus. So as you know, we got two type of diabetes mellitus. In both form of the disease, they also have the same cytotoxic component is by either induced by cytokine as in type 1 diabetes or by elevated level of glucose and fatty acid in the type 2 diabetes. So to find the mechanism response for the cytotoxic effect, this compound usually proteomics approach can be used with a 2D gel electrophoresis and the surface enhanced laser distortion or ionization time of flight mass spectrometry have been undertaken. So that's kind of techniques that can be used to profiling the protein to analyze the disease and the anomaly in the protein level. So in those articles also describe the methods and other methodological aspects of protein profiling. So I think that's all from me, and thank you for having my attention for my presentation. So thank you so much.